Synapse, our online community.
Yeah. So we are live now. You can go ahead.
Hello everyone, welcome to SciComm Synapse, our online community where researchers, students, and practitioners connect to turn ideas into impact. I'm Akash Surve, and we are glad you are joining us live. Today we are speaking with Dr. Abhijit Majumder, professor at IIT Bombay. His lab studies how physical forces and geometry shape cell behavior, mechanobiology, with applications in biomaterials, stem cells, and organ-on-chip systems. Dr. Majumdar's path scans chemical engineering at NIT Durgapur, an MTech in cryogenic engineering at IIT Kharagpur, a PhD in IIT Kanpur, postdoctoral work at INSTEM NCBS in Bangalore and Brigham and Women's Hospital, Harvard Medical School, and a DBT Wellcome Trust Early Career Fellowship. Dr. Majumdar, welcome and thank you for being here.
Yeah, thank you, Akash. It's a privilege to be here.
I would like to request you to you began your journey in chemical engineering and moved into cryogenic engineering and eventually mechanobiology. What drew you through these transitions and how do they connect in your current research? Please explain us.
So I started, as you said, I started as a chemical engineer. Even today, I am a chemical engineer, definitely. The thing is, it's a bit interesting part here is that even from my childhood, when people are in school or so the other people ask them, the relatives ask them, ke, bade hoke kya banna hai? So what do you want to become when you grow up? And my answer generally was, I'm talking about when I was in first standard, second standard. I used to say, I want to become Galileo. The thing is that I did not know the term scientist or researcher, but I read about Galileo and something that I wanted to be. Also, eventually, kind of gradually, I also realized that I like teaching. So that kind of, happened. But then during my 10th, 11th, 12th, I actually wanted to become a doctor. That was then it shifted. That, okay, I thought that, okay, it's a very glamorous profession and also very noble profession. So I wanted to become a doctor. I appeared for the entrance exam first time. I got into engineering, but could not crack the medical. So I did not take up the engineering. I joined, honors in chemistry and continued for 1 year. Prepared for the medical, not for the engineering. Appeared second time. Again, I could crack the engineering, but not the medical. And this time, the engineering rank was even better without any preparation. And that point of time, my sister was doing her PhD at JNU in molecular biology. And I still remembered, so it was the time of December, this time, the Christmas time, she visited. So as you know, she was doing her PhD in, what we call as the general science stream, molecular biology and the genetics. And she kind of convinced me that I should not continue with my chemistry in my BSc, MSc, PhD. That was my plan B. And what she convinced me is that even if you want to continue in research, you can do that through engineering, but go for engineering. And that would make more sense. And he also told me a lot of the horror stories from PhD and stuff. So kind of convinced me that I should go for engineering. So I went for engineering, chemical engineering. So that's my shift. Because I was already in chemistry and I liked chemistry, so I chose chemical engineering without the knowledge that chemical engineering has nothing to do with chemistry. They are completely two different things. But anyway, eventually I liked it. So the chemical engineering was done. Then I went for cryogenic engineering during my MTech from IIT Kharagpur. And there my project was absolutely computational. Although I kind of published paper did well, but I realized that the computational is not my cup of tea. I want to do something, by hand, not just sitting in front of a computer. So then in my PhD, I chose IIT Kanpur for my PhD in chemical engineering. I chose an experimental lab. I did experiment and so on and so forth. Post PhD, I had some good offer to do my postdoc, but then two factors that kind of drove me. Number one, as I told you, I always wanted to become a doctor. So I wanted to go into something related to biology, something that will have a biological, biomedical application. That was the thing. So that was one reason. And the second reason is that I strongly believe that, that point of time and even now, that one should not continue in his or her or their same field where they have done their PhD. In the postdoc, they should look for something different. So these two considerations got me to apply in a more bio or biomedical related field. And thanks to Professor Josna Dhawan at INSTEM NCBS Bangalore, that her lab was a absolute hardcore biology lab. She took someone who stopped doing biology in 12th standard. I mean, that was her greatness. So I joined her lab, continued there. Then, as you said, I went to Harvard Medical School, Brigham Women's Hospital in Boston, worked at Jeff Karp's lab, and then returned again to INSTEM, this time as DB to Wellcome Trust Early Career Fellow. So by the time I already kind of got from my PhD was about the soft mechanics and the microfluidics. In my that kind of makes the connection with what I started doing in Josna's lab and I am doing till now. That is the soft mechanics, and then I realized cell and tissues are essentially soft tissues. They follow the similar mechanics. So there I could kind of connect my PhD journey and my postdoc training into something called mechanobiology. And again, in my PhD, I worked on microfluidics that connect now with my organ-on-chip kind of research. So as I was telling you, I strongly believe one should not continue what they have done in their PhD, but they should look for the area where their PhD expertise can be applied so they can think of to create their own niche that is not a carbon copy of their PhD supervisor, nor a carbon copy of their postdoc supervisor. That should be their own niche. And that is the kind of journey, and that is how my PhD training and the postdoc training came together, what my lab is doing right now.
Thank you very much for clearing a point that how we should choose further research and your own journey from like trying to become a doctor and understanding what a scientist is and reaching a place, a conclusion from feedback from others also. So I would like to further know that what experiences of your postdoc shaped your current approach as a scientist?
So as I told you that the point number one is that understanding the biology, right? I did not have, I had zero understanding of experimental cell culture, molecular biology, and I had no experience of that. Also, kind of the knowledge, right? So obviously that shaped me, shaped my present research. If I talk philosophically or about the broader understanding of the field, I would say I would share two experiences with you and with the audience. I still remember I thought of some idea. I ran to Josna, my postdoc supervisor at NCBS, and I told her, Josna, this is the idea I would like to do. And Josna told me, okay, Abhijit, but why do you think that it will work? And then I pulled things from the literature which I have already studied. I said, okay, literature A says this, B said that said that. If I put all these things together, this will happen. And then Josna told me, so Abhijit, even without doing a single experiment, just by putting the puzzles from the pieces of the puzzle from the literature, you can answer the question. Why should you even do that? Take that leap. You just write a meta-analysis paper. You choose a question which you cannot answer based on the present knowledge that is available, and then you go ahead with that. So I think that is a very important important learning that I got from Josna. And in Jeff's lab, I remember again, I was discussing with the Jeff, and that was Jeff's way of doing it. Jeff used to ask the question, what is the pain point? Pain point, pain, like dirt, pain point. So the thing is that in the whole research, what is that one thing that is the main bottleneck, which will make or break, in your approach? If you do it, how this is going to solve some long-standing question, and what is that long-standing question? So I think that two things that kind of shaped my thinking about the research. And right now I keep on bothering, disturbing, irritating my own lab members, that asking them the continuous question, so what? So I tell them that I will, and the 5 times of so what question will take you to the core of the, core of the actual question that you are trying to address. Sometimes, when we say, okay, so what are you going to study? I would like to see that if this drug will kill the breast cancer cells. I will ask them, so what? So 5 times of that so what kind of leads to the actual question, what in a broader scheme we are trying to address. So I think these two are my learning from Josna and Jeff. You are muted, Akash, you are muted.
Thank you very much. Yeah, as you mentioned that now you are working, your lab is currently working on mechanobiology. So there is a term called mesenchymal cells, MSCs we call mesenchymal. So can you explain about that and why you chose that, and how it helps in organ-on-chip?
So, okay, my lab, so we call our lab as AIM lab, and AIM does not stand for my surname Majumder or Mumbai, where I am. AIM stands for what we are doing, and that is mechanobiology, materials, microfluidics. So these 3 AIM we try to bring together to address other aim, that is mimicking microenvironment. So essentially we try to mimic the tissue microenvironment inside the body. Why that is important? Because just like us, that the way I behave sitting here versus in a pub, versus with my friends, versus giving in a in a conference, versus in a classroom, my behaviors are not the same, right? The core is same, my genotype is same, but what I behave, my phenotype is different depending on the environment I am in. Our cells are exactly the same thing. Their behavior depends on their environment. So when we try to address anything in the lab, better we can mimic the microenvironment, the result that we'll draw from our experiment will be more predictive, more real, more close to what is happening in our physiology. So that is what we try to make. Now, when I say I try to mimic the microenvironment, the tissue microenvironment are made of a lot of the different things, different cell types, the matrix protein, blood flow, other flow, lot of the different things. What we mainly focus on, that how the material or the mechanical properties, that is how soft versus stiff, that may influence cellular behavior. Why that question is important? Because in many diseases, be it cancer, be it cardiovascular diseases, be it kidney diseases, be it arthritis, many diseases, the rigidity changes. And this change of rigidity actually, or change of stiffness, actually influence the disease prognosis. So that is what we try to mimic in our lab. On top of that, we try to bring the fluid flow, and how, because our body everywhere we have the fluid flow. So how the fluid flow can influence the cellular behavior, that we try to study. We don't work only with mesenchymal stem cells. We work with mesenchymal stem cells, cancer cells, diabetic, and various other placental cells, various other different things. But yes, mesenchymal stem cells play a major role in our research area, because it has a huge clinical potential. Number one, it is an adult stem cell, so it can be collected from an adult human being. We don't need the cord blood stem cell or the embryonic stem cells, those neonatal kind of thing. And the second point is that they have a huge potential in regenerative medicine. That is, they have the potential to create bone, muscle, neuron, cartilage, etc. And third thing is that they secrete molecules that has an ability to control our immune system. So for example, say in case of the COVID, we had an immune storm. That is, people were dying because of the high level of immunity. There are a lot of the diseases that is, or the immune reactivity, I should say. Then there are the diseases, what we call as the autoimmune. In fact, I am a patient of autoimmune disease, which say causes rheumatoid arthritis, ankylosing spondylitis, multiple sclerosis, various other things. They are also mesenchymal stem cells have a clinical application to control the immune activation, deactivation, or the immune level. So because of its clinical significance, we use it as one of our model system, and it is very well established in terms of how they react to the mechanical environment. So we know that they do react to the mechanical environment. How and why and what are the pathways, that is something that we try to unravel.
Thank you very much for explaining about mesenchymal stem cells. And as you mentioned about organ-on-chip systems, what are the key challenges and breakthroughs that you have seen in building these platforms?
So for that, probably for the benefit of the audience, I need to just explain the organ-on-chip a little bit. Organ-on-chip is not an artificial organ. Okay, it is not that if I say heart-on-chip, it is not something that can be implanted as heart. Then what is organ-on-chip? Organ-on-chip is a platform where drug testing can be done. Now, why to do the drug testing I need an organ-on-chip? Because, and this is very interesting to know, is that as you know that we do the testing, something called the preclinical trial. That is, say I have a 10 potential molecules for XYZ diseases. I first check them in the drug on a plastic dishes on the cells. When I get some result, I go for the animal model, mouse, rabbit, dog, etc., etc. When I get some result from that, then it goes to the clinical trial. Now, any drug discovery process, this clinical trial, where we actually use the real human, that is the most lengthy as well as most expensive. Millions and billions dollars are spent in that clinical trial phase. Now, the interesting part is that preclinical trial, which is supposed to be a screening process. So in the preclinical trial, if we screen 100 molecules, so they pass the preclinical trial. Out of that 100 molecules which passed, out of the 100, 90 to 95 fell in the clinical trial. It is something like that. You do a whole scrutiny and you book your flight ticket absolutely on time. You reach the airport and you realize out of 100 flights, 95 flights are not taking off that day. That is that kind of fiasco, right? And that is going on, right? That is there. And why this is happening? This is happening because the test that we are doing in the preclinical trial, one is the petri dish, another is an animal. They are not human. Microenvironment is different. And as a result, the result I am obtaining from those preclinical trial that cannot be extrapolated to the clinical trial, and they fail. There is another part of this story. The another part of the story is that, again, remember this 100 molecule that got passed through the preclinical trial phase. To pass this 100, I have already failed 900 more. I started with 1,000. 900 failed in my preclinical. Only 100 went to the clinical. But those 900 that failed in the preclinical trial, they will be never touched in the clinical trial. So So as I already said, my predictive ability is poor. Now the 900, the candidate whom I said, just to imagine, it is something like this. I imagine that there is only one single institute in India to get your PhD degree. And 1,000 students apply, 900 I reject based on the interview. And those 900 will never ever get a chance to do the PhD. But now the 100 who come to do the PhD, my screening process is so bad that out of those 100, only 10 can complete the PhD. Rest 90 cannot complete. That tells my screening process was bad. That also tells me, meaning the 900 that I have rejected, who could not get a chance to do their PhD, probably there were some good candidates, which got rejected and never ever touched for the clinical trial because of my faulty preclinical trial method. So that is where we are trying to do this organ on chip. Now, coming back to your question, the main bottleneck is that what should be the validation protocol? If I say a kidney on chip, what is the meaning of kidney on chip? There is no standardization, whatsoever. Everyone is doing their own way, defining it in their own way. There is no national or international standard. So as a result, the pathways are still not established. While, say, for example, FDA or US FDA or European Union, they are telling that we need to reduce the animal experiment and go more towards this organ on chip kind of models, but the pathways are not yet defined. How the regulatory approvals will happen are not defined. If I want to make say a kidney on chip, what parameters I should study is not defined. So the whole field is still in a very developing phase. So I think that's a bigger philosophical challenge, that if my organ on chip is meaning I am mimicking the microenvironment. When I say mimic, the main question is that what to be mimicked. So if it is like a mimicry, right? If you want to mimic me, you have to catch up some of my mannerism, that if you mimic that, everyone will be able to understand it is Abhijit. You don't want to make a clone of Abhijit. Same of the thing. To catching the points, that what are those critical and the crucial point that should be mimicked, that we need to understand more and more.
Thank you very much. Now we know exactly where the bottlenecks are happening in research and why organ on chip was necessary and how it is going to help. And also that you're working on the pivotal part where your supervisor earlier said that if it's already all clear, then why do you need to work on it?
Exactly.
So you're implementing it.
Yes.
Now we would like to go towards, just a minute. Yeah. So now, as you have mentioned that what were the bottlenecks and you're working on organ on chip, you have also mentioned regenerative medicine. So looking ahead, which applications in healthcare or regenerative medicine excite you the most? And if resources weren't a constraint, what big mechanobiology question would you tackle?
Okay. I will answer your second question. I will not answer the first question. The reason being, again, I mean, it is me, that what excites me most, that keeps on shifting. Okay. Maybe I will give you some answer tomorrow. Someone in the lab will show me some results and I will just get, wow, let's jump into it, right? So that I am a bit fickle-minded in that sense. But yeah, if I can answer one mechanobiology question, what that would be, that I know the answer. See, the thing is, at least, I mean, in the biology, say, for example, we say something as the central dogma. So what is that central dogma? That is DNA to RNA to protein. And that one thing is true, universally true, for any living organism, right? So essentially, the goal of doing science is to find out the universal principle. Today, we say that the Newton, we kind of say the Newton is a great scientist, not because his law can explain why apple falls from the tree, because his laws are applicable throughout the universe, right? So all purpose of the science is to go from the lab to the universe. That is the whole idea. In the mechanobiology at this moment, we do not know the grand unified theory of the mechanobiology, that whether all cell types they sense the rigidity in the same way. There are various mechanical signals like the rigidity, viscosity, shear stress due to fluid flow, vibration, stretching, strain, geometry. Are all these different mechanical cues work via a same pathway? If yes, what is that common thread? Because we see a very absolute commonality, between all of those things. But what connects all of those into a one single thread? And for a cell to finally function, there has to be the gene expression or gene repression, right? Some epigenetic modification. What connects these external or internal mechanical cues finally through the common thread to the epigenetic changes? So that connection, that whole, uh, unified, again, if I'm allowed to use the word, unified grand theory of mechanobiology, that is something that I would kind of, that excites me the most.
Great. So, as like the whole process, what is the flow and from where to where and what connects everything?
Yeah, and like if I can pinpoint, see the again, as I was talking about the the Newton's ones, right? The beauty is that F equal to ma or P equal to mf, whatever. It's such a simple thing, right? The force equal to capital G into mass divided by r square. A very simple expression, but that can explain from sun to the everything, stars to the galaxy to everything, right? So what is the simplest and the most elegant thing to explain the universal mechanobiology? Is there anything that absolutely simple and elegant thing? And obviously I don't know the answer. Obviously the field does not know the answer, but obviously I even don't, and that's the beauty of the doing research, is that I always say the research is like a treasure hunt where you don't even know if the treasure is there or not, right? That's the beauty. So I even don't know if it exists or not. But intuitively looking at every process in the nature, it feels like it should be governed by something very 2 plus 2 4 kind of simple thing. Because everything in the nature boils down to absolutely simple, but simple in that sense, the rule that can be explained in a
very concise and clear.
very concise way. Yeah. So something should be there, I believe.
No, we believe that you will contribute a lot to reach there.
Hope so. If I reach there, probably I will get a Nobel Prize. So I'm not hoping for that at this moment. But yeah, I mean, that is something that one would like to kind of, think.
Yeah. We would love to have you again.
Definitely.
Yeah. So your vision paints a compelling roadmap. Stepping back, how do you see mechanobiology evolving over the next decade in tools, theory, and clinical impact?
So I think that it is going to impact in a big, big way. Okay. And that is, I'm saying, because in the last couple of years, if you look at, say, when we talk about big journals, we talk about CNS, Cell Science Nature, right? And also other, the advanced materials, and the various different PNAs, various
Nature.
different big journals. Almost all of those journals in last 3-4 years published one dedicated issue on mechanobiology. That kind of tells that it is coming there, right? And also the way we have seen, and obviously many other people from the literature we have seen, is that it impacts every aspect of a cell. whether it will survive, it will die, it will migrate, it will replicate, gene expression, secretion. Every aspect of our cell life has been shown by influenced by the mechanobiology. Point number 2 is that regular cell biology are done in the plastic petri plates, which is very unnatural. I have already told you that our plastic petri plate, its stiffer is the gigapascal range, right? It is power 9. Nothing in my body is that stiff. And our brain is essentially like a jelly, that soft. So we are growing the cells and trying to draw the conclusion in a very artificial manner. Obviously, that is effective. I'm not saying that is not effective because we have progressed so far based on that knowledge. However, we have also realized that that has its own limitation. So I believe that soon it will become like that, that the reviewers will start asking question that have you checked the mechanobiology effect? You have cultured the cells on the plastic, fine. Have you checked the mechanobiology effect? The way now people talk about, right? Have you checked the effect in mouse? So that have you checked? So that, say for example, the drug testing, as I was saying, that the drug testing. Now the FDAs are telling that you stop using the mouse experiment and gradually go to the organ-on-chip or the organoid models, right? So essentially, if we need to, what is organ-on-chip? Organ-on-chip is mimicking microenvironment. Can I mimic my brain without mimicking its mechanics? We cannot, right? So I'm sure, so putting all these pieces together, I can envisage that it is going to become an integral part of the doing cell biology, cancer biology, that kind of research. And in terms of theory, as I already said, I see that more and more we will get the pieces of the puzzle that will help us to, again, going towards the unified grand theory. Whether we will reach there or not, or whether the unified grand theory exists or not, I don't know. But I will see that more and more commonalities that we will eventually try to discover.
As you mentioned that, and like nowadays people are also concerned about animals much. They don't want animal testing, so people, we hope that it'll become a standard, that mechanobiology testing will become a standard for everything. And progress also depends on inclusive teams, and you are one of the few openly LGBTQ plus scientists in India. How has being open about your identity shaped your journey in academia?
I actually don't think that it has, I mean, the being open or not has shaped my journey in any way. Okay. Also, my coming out has always been very gradual. It was not like one day I announced it. It was very gradual. And no, I don't think it has shaped my journey per se. No. I mean, obviously, it has shaped my personality. There is no doubt about that. I believe that it has made me more tolerant to other views. It had my sexuality had made me to understand the privilege and underprivilege, both the positions. It has helped me to understand the intersectionality. That is that a person can be privileged in many other aspects, but still being underprivileged in one aspect may have a huge impact. So those understanding probably helped me. But yeah, I think that's it.
Yeah, thank you for sharing that. And for many LGBTQ plus students who dream of STEM, they do hesitate to be open. What practical advice would you offer to them?
Number one, everyone's journey is different. We all come from very different backgrounds, right? There is no rush to come out. Do not get under peer pressure to come out. Your family, I mean, the point number one, unless you are absolutely certain about your family support, till you are financially independent, you don't need to come out. I mean, okay, if that gives you a mental peace, That's your choice. But again, I mean, what I'm trying to say, everyone's journey is different. Don't come under the peer pressure that, okay, I have to come out till the point you are financially independent. And you are sure, and or you are sure about your family support, right? That's point number one. Point number two is that, again, it depends on, say, even after being financially independent, it also depends on in which workplace you are. Suppose I am at IIT Bombay. I know that IIT Bombay is a open liberals, place where I will not get a backlash. I don't know if I will not name, but if I was at XYZ place, maybe I had to think 10 times before, coming out. So my practical advice is that weigh your pros and cons, weigh the reality around surrounding you. But at the same time, many of the times we have imaginary fears. We just imagine the backlash. That also not true, right? I mean, at least again, that is my own experience, that people surrounding me, they were least bothered about my sexuality. So yeah, that's it. So you have to weigh your surrounding and take a call.
Thank you very much for sharing that. Mentorship and visibility influence how students plan their paths. Many wonder whether to go deep or explore broadly. How should they approach that balance?
That's a very, very good question. Very good, excellent question, because okay, in fact I will say that I feel when I started my independent career as a PI, somehow I now feel, now when I look back, I feel like I have broadened too much compared to the depth. That is my own kind of own assessment after 11 years of my. Probably I should have broadened a little less so that I could. Because when I broadened my field, what happened is that in my lab, 10 people are working in 10 different areas. As a result, I need to get 10 types of reagents to kind of, support them. And each one of them progress this much, but they don't get add up. They all are, so that is one thing. So I mean, I said it's an excellent question, but I really also don't know how to answer it. That, I mean, a very cliche answer would be, don't go too deep, don't go too wide. But what is too deep and too wide is a difficult question. Let me think over it. Let's go to the next question. If something comes to my mind, I'll come back.
Sure, sure. Failures are often part of scientific life. Could you
Can you
share a
please
moment
repeat it?
when, yeah, failures are
Failures,
often
right?
part of, yeah, part of our scientific life. Could you share a moment when something didn't go as planned and what you learned from it?
I mean, that is not part of life, that is the life. Scientific research is the failure, right? It is always, as the famous quote is there, I did not fail 999 times. I have learned 999 times how it should not work.
Does not work.
How it does not work. So, yeah. So I think there is nothing called failure in research per se, I will say. I always say it is deferred success. It is success deferred. In fact, I was in a governing body in our campus school, and there I proposed that stop giving fail grade till that deferred success, deferred pass. So fail. So okay. Now but yeah, definitely it gives you a moment of feeling bad, moment of rejection. That definitely happened. I will not say that does not happen. And I mean, you were asking me the example. I mean, there are too many examples, right? When some paper you had a high hope, it got rejected again and again. Day before yesterday, one of my project proposal got rejected, and comments were like, very, very. I mean, I said that they said something that was even not there in the proposal. So yeah, so but then that is the part of life, right? That is the part of life to go ahead with. Uh, Most of the time, the I think the way to go ahead is always celebrate the small success, particularly in STEM and the research. But I will say even in your general life, right? I mean, there are many, many. In fact, see the okay. I'll just tell you one thing. Last month I was in October I was in Sweden, and in a relative's place, and I saw in a kindergarten school a tiny kid, probably 3 4 years old, and there was a pool of mud, a puddle, and there this guy was simply jumping, doing nothing, just jumping and laughing, jumping and laughing. There was no one seeing him, no one is cheering. He himself was jumping and laughing. And I was telling my relative, at what point we lost this joy? This is a pure joy, right? You are not waiting for someone to cheer. You are not waiting to get anything back. It is just you are doing something and getting, splashing the water, and that is giving you the joy, happiness, right? So that is what that may be doing one experiment nicely. Celebrate that. Getting, one troubleshooting that the antibody was not working and finally it is working. Celebrate that. So celebrate those tiny, tiny successes as well as as I say, in your own personal life. I mean, we all have the personal moments all the time. So we should celebrate that all the time. And again, something that I tell is that if we think of a life, right? Uh, Life, I feel like a necklace with beads and strings. String is the time, which is invisible. We cannot see it, right? You do not remember what you ate 3 days ago in lunch or dinner, but you remember a moment 15 years ago. So these beads are the moments. String is the time, beads are the moment. That creates the necklace. What we see are the beads. So just cherish those pearls of moments. That's it. Rest it will take care of.
Thank you very much. We will always remember that we have to enjoy what we are doing and be more satisfied in that, and then look
I
at
mean,
the
I will say that
results.
even you may not enjoy what you are doing. You may not enjoy at all. That's okay. If you have to repeat an experiment 50 times, I don't expect you will enjoy to doing that 50 times. But I'm saying after the 50th time when you get the result,
Mhm.
celebrate that moment.
Yeah,
Give reward to your mind. Our mind crave for rewards. Give rewards to your mind.
that's very insightful.
That reward can be an ice cream candy or a movie
Yeah,
that reward
yeah,
yours.
yeah. Sure. Today's session will go well, and I will go watch a movie today.
Good, great.
That's very insightful. I think towards the close, do you now got an example about the question of too wide, too deep, or should we go to the next
Ah,
last question?
deep and right. Yeah. I think I have already actually given the answer in the next process, right? That
Yeah,
go ahead till the point you are getting satisfaction and you are getting
yeah.
kick out of it, right? Uh,
Mm.
and I think that's it. I mean, there is no unified grand theory
Clear
of
formula.
happiness, right? And life does not have a unified grand theory. It just goes on.
Yeah, that's
And
so.
also, something that don't take yourself too seriously. I mean, we are not that important in the big scheme of the universe. So let us not take us that seriously.
So to close, what is one guiding principle you would like students and early career researchers to carry with them in science and life? You have already given us many life lessons. But still, like, finally, one thing that we should surely remember from this session.
So I will probably continue what I just said and kind of, but we'll tell it in a little different way. That we all know that shlok of Gita, right? That the karmanni vadhikaraste ma phaleshu kadachan. We often take that in a little different meaning. We say that don't expect the outcome. That is not true, because if Arjun throws his arrow without aiming, just okay, I don't care, where it goes, he would not get the success. His aim was to get win in the battle, right? It was there, definitely. So there are the goals uh, Arjun also had. So karmanni vadhikaras, the math sort of, when I say that don't expect the outcome, that's wrong. You have to expect the outcome. You have to plan for the outcome. Then what is the meaning of that statement? The meaning is that don't get attached. Don't get attached to it. As I said, don't take it too seriously. Particularly in science, that's a very interesting thing, is that when you are doing the work, that point of time, you have to care it as if it is your baby. You have to be fully attached. As soon as the experiment is done and you are analyzing your data and verifying your hypothesis, you have to be completely detached. You have to be ready to accept that your initial hypothesis was wrong and you were stupid to think of that hypothesis. Right? So this constant attachment and the detachment is important. That is, again, I will say, in the personal life. That, again, I will give you a very, very, very personal thing from my right now. My parents are 87. Both are 87. They are staying, they live with me. And as 87, they don't keep that way, right? When I say the attachment and the detachment is that I'm trying to do my best to keep them healthy. Any small thing, I will consult the doctor. I will try to keep them whatever possible, both in terms of their physical health, but as well as their enjoyment, whatever they would like to do as their hobby to keep them engaged with painting, drawing, etc., etc. That is attachment. Detachment is that I know they are 87. They have done a full life. I know I cannot change the obvious. So keeping myself mentally prepared for the obvious, it will always be a downward hill. At 87, no one's health improves. It will always be a downward hill. I cannot feel dejected seeing that. I have to say that my job is to give them the best life, whatever years, days, months they have. I have to give them the best life. At the same time, I have to be ready for the eventuality. So I think that's the very personal thing that I have shared. And I will say that if you can get into that frame of the mind, you can be happy. Whether you can be successful or not, I don't know, but you can be happy.
Thank you very much for sharing the moments and understanding. We have some YouTube questions. We'll take them quickly.
Sure.
What are the unpublished and unwritten challenges while working with mesenchymal stem cells? Mesenchymal stem cells.
Unpublished, unwritten challenges. I mean, this is very, I don't know what the person meant, but I can say about the challenges. Challenges is that number one, if I talk about the adult stem cells, so they are the primary cells, and you may know that the primary cells have a certain lifespan, that I can grow only up to this point. After that, they lose their property or they gain some unwanted property. But the number of cells that we get, that is very limited. But for the application, I need a huge number of cells. So going from that limited number to that big number, you need to do the cell multiplication. But the problem is that if you do the multiple times cell division, they lose their potential. So I think that is one of the main challenge. Another challenge is that, and that is from the various, so the mesenchymal stem cells are going through various clinical trials. One of the major issue, and very recently actually FDA approved the first ever MSC based treatment strategy. But one of the challenges is that there is a huge variability. Mesenchymal stem cells collected from one donor to the other donor to the other donor, there is a huge variability. How they are very finicky in that sense, how you treat them, they behave accordingly. So as a result, having the reproducible standardized result is a big challenge.
And the next question is, how can we understand mechanobiology from computational angle of biology?
I think that is again another very good question. And as I was telling you, we are talking about the unified grand theory, right? We cannot reach the unified grand theory without the computational angle of biology, right? Because we have to simplify what we are seeing. We have to find out that common goal. So I think it will be kind of a feedback that what we understand from the mechanobiology, we have to give to the computational as well as the system biology. They will put it in their model, then they will predict something. We will again try that. So they are, and I believe that almost every field of biology right now, not just the mechanobiology, will be benefited greatly from the computational angle of the biology.
Soumila, you want to say something?
Yeah, one small question I have regarding mesenchymal stem cells. Sir, do you any experience the problem of JVHD when you are working with mesenchymal stem cells?
I have not worked with, I do not work with or what of the GVHD, but as you have correctly said, so graft versus host diseases. So their mesenchymal stem cells are being in the clinical trial for the various different, at different phases of the clinical trial by various different entities. But my lab does not work.
And secondly, sir, mesenchymal stem cells require feeder layer of cells to grow again.
No, that's not correct actually. The adult mesenchymal stem cells, or even the from the cord blood, they don't need, the embryonic stem cells, they need the feeder layer. Mesenchymal stem cells, they don't.
Oh,
so that's actually a benefit of using again the mesenchymal stem cells. Advantage.
Okay, sir. Thank you, sir.
Thank you. Good questions.
Yeah, one of my last question, but it's a short of time still, I want to ask that how difference have you observed in different institutions? You have worked across countries and different institutions. So what was the feeling of it? Like, what is the culture? What people are focused on, or whatever you felt different?
I actually get this question quite often. And the problem in answering the question is that at different institutes, I was at different roles, right? And at a different time point of the society has changed from my BTech days, 98 to 2002, to now to a way that, if someone time travel from 98 to now, they will not be even able to understand, what is going on. Someone is paying by using the phone. Even the phone was unseen that point of time, right? So I think, or like we are talking, that was even unimaginable in my college days. So everything has changed. So it is very, very difficult to compare. Still, if I have to compare, I will, or kind of, I will say that IIT Bombay, being in the metro city, has a lot of the things to do outside, right? Very easy to go outside, which was not there when I was in, say, Kanpur or Kharagpur, comparatively. Even in Bangalore, INSTEM was so far away from the main city that going to the city was we need to plan that, okay, today we will go to the city. So I think that overall changes the way we function, because in those places, because there was nowhere to go, we were more cohesive, which I see a bit less here because we have a lot of the things to do. On the other hand, the here again being in the metro city, the overall outlook is comparatively more progressive and liberal. But again, I would say that I'm comparing at a different time point. I don't know what was the time point there and now, right? So as I say that I don't know how the mess food of the student hostel taste in IIT Bombay. How can I compare?
Sure. Thank you very much. So, to end the session, I would like to thank Dr. Abhijit Majumder for a compelling and generous conversation. Your insights into mechanobiology, biomaterials, and organ on chip will inspire our community. And
Thank you, Akash.
majorly
It
your
was fun.
life lessons also.
Yeah, it was fun to talk to you. Great.
I would like to thank our audience for joining and engaging, and to the Science SciComm Synapse team for smooth online support. We would really appreciate your feedback to make our next event even better. The feedback form link is in the comments chat. Please submit it to help us improve and to receive your e-certificate. On behalf of SciComm Synapse, thank you all and see you next time.